What's up, everyone? Today I'm going to walk you through aneuploidy and polyploidy. This is one of those core genetics topics that shows up everywhere, whether you're studying in clinic or just trying to understand how chromosomal conditions arise. So let's start by building a foundation. Now, when we talk about chromosome number, we're really talking about how many complete sets or pieces of genetic material a cell has. So first, we have a haploid number, which is what we see in gametes like sperm and egg, and this is represented by the letter N. There's also a diploid number, which is what we see in most of our body cells. This is represented by 2N. Now from there, we define two important concepts. A euploid number means you have exact multiples of the haploid set. So 2N, 3N, 4N, that's an even organized system. But when that organization is disrupted, we get two main categories. The first is polyploidy. Polyploidy is when there are extra full sets of chromosomes. And the second is aneuploidy. Aneuploidy is when there are extra or missing individual chromosomes. And that distinction is really important because it directly impacts how severe the condition is. So, why does this matter clinically? One of the most important things to understand is that chromosomal abnormalities are actually very common, but they're not often compatible with life. At birth, we see chromosomal abnormalities in about 1 in 110 individuals, and autosomal trisomies in about 1 in 700. But if you look earlier in pregnancy, especially in miscarriages, over a quarter involve trisomy, and nearly 10% involve polyploidy. So what this tells us is that many chromosomal abnormalities occur, but only some make it to live birth. Now let's start with polyploidy because it's conceptually simpler. Polyploidy means you have extra complete sets of chromosomes. So instead of 46 chromosomes, you can have triploidy, which equals 69 chromosomes, or 3 complete sets. You can also have tetraploidy, which equals 92 chromosomes, or 4 complete sets. Now here's the important teaching point. When you duplicate entire sets of chromosomes, you're disrupting the balance of thousands of genes at once. That's why polyploidy is usually not compatible with life. Triploidy occurs in about 1% of conceptions, but only about 1 in 10,000 live births. So again, it's very common early, but rarely seen later. Now, how does triploidy happen? The most common mechanism is dispermy, meaning that one egg gets fertilized by 2 sperm. So if each sperm brings 23 chromosomes and the one egg fertilized brings 23, you end up with a total of 69 chromosomes. Other mechanisms include a diploid sperm or egg due to meiotic error, or failure of the egg to properly discard extra chromosomes, like a polar body issue. Now, a high yield fact to know is that the parental origin of the extra chromosomes matter. If the extra chromosomes come from the father, you tend to see abnormal placental growth, and this is called diandric. It sometimes resembles a partial molar pregnancy. If they come from the mother, you tend to see severe fetal growth restriction. This is called digynic. So there's the same chromosome count, but very different presentation. Now let's take a look at tetraploidy. Tetraploidy is a bit different. Instead of a fertilization issue, it usually happens because the cell divides its DNA but doesn't divide it into two cells. So now everything is doubled. This typically happens very early in embryonic development, and it's almost always not compatible with life. Now let's shift into aneuploidy. This is the most clinically relevant category. Aneuploidy means that there is one extra or one missing chromosome, not a whole extra set. And the main mechanism behind this is non-disjunction. So what is non-disjunction? Non-disjunction means chromosomes fail to separate properly during meiosis. To understand this, think about meiosis as a sorting process. In meiosis 1, homologous chromosomes separate. In meiosis 2, sister chromatids separate. Now, if something goes wrong in meiosis 1, non-disjunction, both homologous chromosomes go into the same gamete. If something goes wrong in meiosis 2, non-disjunction, sister chromatids fail to separate. Either way, you end up with gametes that have too many chromosomes or not enough chromosomes. And when that gamete is fertilized, you either get trisomy, which is an extra chromosome, or monosomy, which is a missing chromosome. Now, what if everything goes normally at fertilization, but an error happens after? That's when we get mosaicism. This means that different cells in the body have different chromosome numbers. So instead of every cell being trisomic, only some of them are. And clinically, that often means the features are milder or more variable. Mosaicism can happen through mitotic non-disjunction or anaphase lag. Now, why does aneuploidy happen? The biggest risk factor is advanced maternal age, and here's the reasoning behind that. Egg cells begin meiosis before birth and remain paused for decades. Over time, the machinery that separates chromosomes becomes less reliable and is more prone to errors. There are also associations with reduced recombination and possible genetic predispositions. But importantly, there is no strong evidence linking lifestyle or environmental exposures to non-disjunction. Now, across different trisomies, there are some consistent patterns. Why is that? It's because having an extra chromosome disrupts gene dosage across many systems. So we can often see growth restriction, developmental delay, congenital anomalies, and dysmorphic features. This reflects how tightly regulated development normally is and how sensitive it is to imbalance. Now let's walk through the big three autosomal trisomies. First, we have trisomy 21, or Down syndrome. This is the most common viable trisomy. So when you hear trisomy 21, think hypotonia, flat facial features, upward slanting palpebral fissures, a single palmar crease, and congenital heart defects, especially an AV canal. Next, we have trisomy 18, or Edwards syndrome. Here I always tell people to picture the hands, which have clenched fists and overlapping fingers. Individuals with trisomy 18 also have a prominent occiput and growth restriction. This condition has a high mortality rate in infancy. Lastly, there's trisomy 13, or Patau syndrome. This one is all about midline development. So think holoprosencephaly, cleft lip or palate, polydactyly, and scalp defects. It's also associated with severe outcomes and early mortality. Let's take a look at this information from a counseling perspective. From a genetic standpoint, most cases of aneuploidy are sporadic and due to non-disjunction. Recurrence risk is typically low, but slightly increased compared to baseline. An important exception is translocation-associated trisomies, which can significantly change recurrence risk. So always consider the mechanism, one's family history, and parental karyotypes when appropriate. Now let's put all this information together. If you step back, the big picture is this. Polyploidy disrupts entire chromosome sets, so it's usually not compatible with life. Aneuploidy affects individual chromosomes and is sometimes compatible with life depending on which chromosome. Non-disjunction is the main underlying mechanism of aneuploidy, and maternal age plays a major role in risk. And that's your review. Hit that like and subscribe button for more quick study tips, and I'll see you next time.